Okay, yeah, so as I said, I'm Carol Lee from the University of Wisconsin, and I'm really happy to be here. And I'm going to be talking about recent work from my laboratory focusing on genomic mechanisms underlying or promoting parallel polygenic adaptation during rapid habitat shifts. And I'll be talking both about climate change and invasive species. And the mechanisms involved are quite related, and you'll see why pretty soon. So as you all know, populations throughout the globe are encountering unprecedented rates and levels of environmental change. And due to increases in ice melts and changes in precipitation patterns, climate change is driving drastic alterations in habitat salinity throughout the globe. So if you see in these blue regions, this is where salinity is declining. And if you see in these darker, warmer areas, salinity is increasing. So we're getting a dramatic shift in the global water cycle. In addition to that, many invasive populations are undergoing fundamental niche expansions. They're going into novel salinities. For example, if you look at the Great Lakes of North America, they're currently overrun and dominated by brackish water species that you might be familiar with, like zebra mussels and quagga mussels, and a lot of brackish water amphipods. In fact, if you look at the numbers of invaders in the Great Lakes, they're heavily enriched by brackish water species, far more than you'd expect based on expectations based on propagular pressure and the abundance in the native range. So there's a disproportionately high number of brackish water species invading freshwater habitats. Now, this phenomenon is quite unusual when you think about biogeographic patterns, where this intermediate salinity of 5 parts per thousand tends to divide the biogeographic boundary between saltwater and freshwater species. And it's very unusual for species to cross this sort of impenetrable biogeographic boundary. So the question then is, what accounts for the ability of some populations to invade novel habitats when most of them cannot? So this question is related to the issue of evolvability of populations and the nature of their evolutionary pathways. So when you think about these evolutionary pathways, a key question is, to what extent are these pathways deterministic? And you're probably familiar with Stephen Jay Gould, who once said or asked, if we rewind the clock and replay the history of life on Earth over and over again, do we repeatedly get the same outcome, or do we get different outcomes? And Stephen Jay Gould argued that because of historical contingencies and accidents that would play a significant role, he argued that replaying the tape of life would lead to evolution down very distinct evolutionary pathways each time. So he argued for non-parallel evolution as being widespread. Well, what do the empirical data say? So if you look at single gene studies, it's really hard to read from here. Many single gene studies uncovered striking cases of parallel selection acting on the same loci, and you're probably familiar with some really nice case studies on that. However, when you look at multigenic traits encoded by many genes, polygenic adaptation has typically revealed non-parallel evolution, with selection favoring different alleles among replicate lines. So the argument there is genetic redundancy would lead to alternative evolutionary pathways because you can achieve the same trait optimum through different beneficial alleles. So if you look at theoretical predictions, such as polygenic shift simulations, as the numbers that encode a trait increase, you would predict that the degree of parallelism would go down. And this is because many of the loci would be functionally equivalent or redundant, allowing many different evolutionary pathways to occur. So that is the null expectation. The null expectation is that polygenic traits should display genome-wide patterns of non-parallel evolution. Now, if you look at some key studies using the fruit fly Drosophila melanogaster in response to temperature adaptation, you do see patterns that fit this null model of non-parallel evolution among replicate lines. However, when you look at the factor temperature, it has global effects on physiology, affecting all biochemical reactions and all metabolic processes and ectotherms. So you would expect temperature adaptation to be non-parallel, given that it's an extremely polygenic, temperature response is extremely polygenic in its response. So what happens when you look at other physiological traits in other systems? And what are some important factors that might affect the extent of parallelism? So some of the key questions regarding polygenic adaptation are as follows. What if the loci are not redundant and not interchangeable, which is the assumption? What if some alleles and loci are absolutely necessary for physiological adaptation? And what is the role of population genetic structure, such as the amount of standing genetic variation in the population? And how might genome architecture affect the trait under selection? For instance, the number of loci might be important, the linkage among loci, and the interactions among the loci, such as epistasis. So answers to these questions are critical for understanding how populations might respond to rapid environmental change, as the physiological responses themselves tend to be polygenic. So to address these questions, my laboratory is using the charismatic microfauna, the copepod. How many of you know what a copepod is? They're really important. Most abundant animal on the planet, likely. And we're looking at the Eurythemra affinis species complex. And this particular species is incredibly important for supporting many important fisheries throughout the world, such as herring, salmon, anchovy, et cetera. So the native distribution of this charismatic copepod spans coastal regions in estuaries and salt marshes in North America, Europe, and Asia, shown here in pink. However, within the past few decades, due to ballast water transport, this copepod has invaded many freshwater reservoirs and lakes throughout the world, shown here in blue. So where did these populations come from? So when we take a phylogeographic approach, we see that the freshwater populations, shown here in white, invaded multiple times independently from genetically distinct clades represented by different colors in different geographic regions throughout the world. For example, there are multiple independent invasions in the red clade, in the Atlantic here, and multiple independent invasions in the green clade, the Gulf clade, shown here. And I'm going to show you these clades over and over again. There'll be a test at the end, don't worry. So when we look at this invasion that occurred in the St. Lawrence estuary, this occurred only about 60 years ago. What is the pattern of physiological evolution that occurred from the saltwater population into the freshwater population? So to address this question, we perform common garden experiments. And I'm showing you this design here because all the subsequent experiments have this design. So when I show you microarrays, genomic signatures of selection, laboratory experiments, it's all this design. So what we do here is we take the ancestral saline population and the freshwater invading population, and we first rear them at a common salinity to remove the effects of environmental acclimation. Then we take individual egg clutches representing genotypes from each population, and we reared them across a range of salinities to look at the physiological response across these salinities. So what is the pattern of physiological evolution? So if we look at the ancestral saline population from the St. Lawrence estuary, the mean physiological response, here you show survival to adulthood. We have low survival under low salinity conditions and higher survival under higher salinity conditions. What about the freshwater population, which is only 60 years old? Well, we see the opposite pattern of a high survival under freshwater conditions and low survival under higher conditions. This is a really rapid response occurring only across a few decades. It's an evolutionary response since this was based on a common garden experiment, which removed phenotypic plasticity. So given this dramatic shift in physiological response, what is the particular trait that's undergoing natural selection that's allowing these freshwater invasions to occur? So a great challenge for a charismatic little copepod like Eurythemra affinis is that as environmental salinity goes all the way down to zero, the organism needs to maintain an elevated hemolymph osmolality, or blood concentration, relative to the environment. So this is the pattern for the saltwater population. What would the freshwater population look like? Would it be higher or lower than this line? What do you think? Well, the answer is that the freshwater population, which is only a few decades old, shows an elevated hemolymph concentration relative to their saltwater ancestors. Across these independent invasions, you get the same pattern. And what this means is in the freshwater population, you get an evolution of increased body fluid regulation, increased homeostasis relative to the environment. And we see this pattern a lot in ectotherms. And what this means is to maintain this higher blood concentration in the face of a hostile freshwater environment, the organism needs to either increase ion uptake from the environment or reduce ionic losses. So how would this take place? Where does ion uptake take place, and how would this trait evolve? So the action is in the ionocytes. Sorry, I'm going to show you some physiology now. So the ionocytes are specialized cells that are embedded within osmoregulatory organs like gills and kidneys. You have them too. And they're enriched with ion transporters that perform ion uptake and ion excretion. And I argue that the evolution of ionocyte function must be critically important for crossing these major biogeographic boundaries. So how would this occur? How would ion uptake take place? This is controversial. This is my model right here, our model. This is the fish model, but it's not really resolved. And a lot of these ion transporters are very recently discovered, and I'll show you that in a minute. So the great challenge is taking up ions from a freshwater environment against a concentration gradient is extremely challenging. So to overcome this concentration gradient, V-type ATPase, the proton pump, would pump out protons into the environment. Then using this proton gradient, other transporters could transport sodium and other essential cations into the cell. And what this sodium transporter is controversial and unknown. And I'm arguing, based on our studies, that it's the sodium hydrogen antiporter, which was only discovered in human kidney in 2013. So this is a new gene. But I'm arguing that it's this gene right here. And then sodium would then enter the hemolymph through sodium potassium ATPase. In an alternative model that was proposed for fish, an ammonia transporter would transport out ammonia. Then this ammonia would react with protons to create ammonium ion. Then this consumption and deficit of protons would force the sodium-hydrogen exchanger to export out protons and bring in sodium, this necessary cation, into the cell, and then sodium would enter the hemolymph through sodium potassium ATPase. Again, these models are not well understood, well resolved, they're controversial. A lot of these ion transporters, are newly discovered, but these are the current models. So the question is, which model would apply here, which one would predominate, which ion transporters are involved, and how would their functions evolve during these salinity shifts? So in order to address this question, my lab performed genome-wide gene expression of the saltwater and freshwater lines under common garden conditions, again, common garden, and we looked at the changes, the evolutionary shifts in gene expression, genome-wide. And what we found were thousands of genes showing differential expression between the saltwater and freshwater lines under common garden conditions. And this is one of the experiments. Showing to the right are the genes that showed elevated expression in the freshwater lines. Toward the left are elevated expression in the saltwater lines. And what we found that ion transporters dominated as the functional category that showed evolutionary shifts in expression. And if we map those results onto the model of ion transport, we saw that the genes involved in model 1 showed evolutionary increases in expression in the freshwater lines under freshwater conditions, suggesting that the loci here contributed to freshwater adaptation, gave support for model 1. And what's interesting is we're looking at these genes in more detail. And when we look at the individual paralogs, we found that they're expressed mostly in the legs. So we discovered that the copepod legs are the osmoregulatory organ. And developmentally, legs are homologous to gills, and these guys don't have gills. So we found them in the legs, and interestingly, we found that different paralogs of the gene family showed very distinct patterns of gene expression, differences in localization, and differences in the amount of expression. We were surprised at how different this was. So going back to our original question, I'll address it in a minute, but with respect to this particular loci under selection, ion transporters dominated as the largest functional category that showed evolutionary response to salinity change. And what was interesting is that the different ion transporter paralogs showed distinct evolutionary patterns and likely have distinct physiological functions. So these ion transporter paralogs, the loci under selection, are likely not interchangeable and not redundant. So what does that mean for polygenic adaptation? So given that these loci under selection, the dominant category, appeared to be non-interchangeable and non-redundant, what does this mean in terms of parallel selection acting on these loci? Do we see parallel selection acting on the same loci and SNPs in wild populations during independent invasion events? And as I mentioned to you earlier, the theoretical models suggest the null expectation is that polygenic traits should display genome-wide patterns of non-parallel evolution based on theoretical null expectations. So to address that question, we examined genomic signatures of selection across independent invasions in the wild populations, in the Atlantic clade populations shown in red, and in the Gulf clade populations shown here in green. And in order to examine signatures of selection, we performed whole genome sequencing for 100 individuals per population. And what we saw were incredible parallel shifts in SNP frequencies across these independent invasions, in the Atlantic clade here in red, and in the Gulf clade shown here in green. And here, the more saline populations are in the darker colors, the freshwater populations are in the lighter colors, and these individual gray lines represent individual SNP frequency shifts, and the diamonds represent the mean frequency for each population. Now, this level of parallelism was incredibly high. Of the 7,000 or so SNPs showing signatures of selection across any of the invasions, 3,000, or 42% of them, showed parallel selection across all the invasions. This is much higher parallelism than you'd expect. And of those SNPs under selection, 350 of them also showed an association with salinity. So when we look at these SNPs under selection in more detail, many of these SNPs under selection and associated with salinity, again, occurred at or near ion transporter genes. And this was similar to the result I showed you earlier for the gene expression analysis, where ion transporters, the same ion transporters that showed differential expression, were also under selection in the wild populations, such as the enigmatic sodium-hydrogen antiporter. In fact, the highest density of SNPs under selection resided within this genomic region containing the 7 paralogs of the mysterious sodium-hydrogen antiporter, with these blue dots representing parallel selection across all independent invasions, and the size of the dot representing the association with salinity. And so, again, this ion transporter gene family is showing up again. And what was remarkable about this is many of the SNPs with signatures of parallel selection and an association with salinity here also showed signatures of balancing selection in the native range population, the exact same SNPs. So I was really surprised by this. So these are the 4 native range populations of the freshwater populations. And what this result indicates is that ancient polymorphisms in the native range could provide the genetic substrate on which selection can act during the repeated invasions. So this is a reservoir in the native range of alleles at elevated frequency that could serve as the substrate for selection, promoting parallel selection during these invasion events. So in terms of population genetic structure, balancing selection in the native range populations and selection on intermediate alleles could potentially promote parallel evolution. So this could be an important factor promoting these parallel events. So in order to more rigorously examine the mechanisms that drive parallelism, we performed a laboratory evolution experiment. And as I mentioned to you earlier, the null expectation is that polygenic traits should display genome-wide patterns of non-parallel evolution. That's the null hypothesis based on theory. So then we focused on the Baltic Sea, where a lot of climate change effects have led to the freshening of the Baltic Sea. So over 50 years, the Baltic Sea has already become much fresher, shown here in blue, due to increases in precipitation and runoff. And the Baltic Sea is expected to become much fresher in the future, perhaps becoming a freshwater lake. So this is really bad. So we took a Baltic Sea population from the saltwater range from Kiel, and we reduced salinity across for 10 replicate lines for 10 to 20 generations. So we took the saltwater population and made salinity go down in this laboratory evolution experiment. So in response to laboratory selection, a certain set of alleles rose rapidly. So the beneficial alleles rose very rapidly during the experiment. So these are the experiment-selected SNPs. In contrast to that, in the control lines, there was no significant change in SNP frequency. And the SNPs under selection were distributed widely across the genome, and this is one of the 4 chromosomes for this species. And again, we see the same loci under selection, the same loci in the gene expression analysis, the same loci in the wild populations are under selection here as the top gene ontology category under selection. And again, you see the enigmatic sodium-hydrogen antiporter under selection here. So the same loci are under selection. And interestingly, across the 10 selection lines, we saw an incredibly high degree of parallelism. This is parallelism in terms of pairwise similarity score. And in our data, about 80%, we saw 80% overlap in the selected alleles between the 10 replicate lines at generation 10. This was incredibly high, much higher than the expectation. The null model expectations here, the multiplicative model, where all the alleles are acting independently. So we got a much elevated signature of parallelism. So then we had a hypothesis, and our hypothesis was as follows. We thought that positive epistasis might be driving this high degree of parallelism that we observed in our experiment. And in this case, multiple beneficial alleles would be selected together. Sorry, one second. And this hypothesis was consistent with the fact that many of these ion transporters would have to cooperate to accomplish the role of uptake from the freshwater environment. They need to work together. So to test our hypothesis, we did a bunch of simulations. We compared our actual empirical data to different models. So we simulated our data under different models of epistasis, such as positive epistasis, negative epistasis, et cetera. So when we compared our empirical results, shown here by these brown lines again, and we looked at the different models of epistasis, only the model of positive epistasis, positive synergistic epistasis, could match the degree of parallelism we saw in our data. So if we look in more detail here, positive epistasis, where the effect of an allele would be increased by the presence of other alleles. Or if you select on a beneficial allele, you're going to select also on other related beneficial alleles. This was the only model that can account for the greater parallelism that we saw, greater than expected, relative to the multiplicative model, the null model, where all the alleles are acting independently. So this was pretty striking. Now, we wanted to look at other factors, because other factors could be involved as well. So we looked at the effect of physical linkage among loci. And we found that increasing linkage among the loci did not increase the level of parallelism. So we decided that was likely not as important. Now, we wanted to look at the effect of standing genetic variation, because as I told you earlier, in the wild populations, the native range populations had higher levels of standing variation of the SNPs under selection. So when we looked at the effect of greater starting frequency, we did see that selection on balanced polymorphisms did promote the degree of parallelism, especially in conjunction with the effect of positive epistasis. So together, balancing selection and positive epistasis could contribute to the level of parallelism. But balancing selection could not alone explain the high degree of parallelism we saw in our data. So as I told you earlier, according to theoretical predictions, as the number of loci encoding a trait increases, you would expect the degree of parallelism to go down. Now, when we look at our empirical data involving 5,000 SNPs on 121 haplogroups, this is very polygenic. Yet we see that the degree of parallelism matches that of only 20 loci under selection. This is a signature of epistasis. And this result is consistent with positive epistasis acting among loci, given that if alleles are functionally linked, they would be expected to behave like fewer loci under selection. So this was a pretty striking result here. And we found that our loci in the lab were of ecological relevance. If we look at the SNPs under selection in the lab, they showed a striking signature of selection relative to the background loci in the Baltic Sea populations. So this is the background loci in 8 Baltic Sea populations. So the experimental selected SNPs showed signatures of selection in the wild, and what was interesting is even though we started with a saltwater population down in Kiel, these experimental selected loci were more common in the more freshwater regions of the Baltic Sea, which was really interesting. So in terms of genomic architecture, it appears that positive synergistic epistasis might play a really important role in promoting parallel adaptation. So to conclude, we find that parallel selection acting on the same loci and same SNPs in the wild populations and in the laboratory selected lines, even though we're dealing with a polygenic trait. And our results indicate that this parallelism was likely driven by positive epistasis and to a lesser extent by selection on standing genetic variation. So this potential role of positive epistasis is consistent with mechanisms of ion uptake, which requires the action of multiple cooperating ion transporters to accomplish the role of ion uptake from a freshwater environment. So it makes sense that very specific, non-redundant combination of alleles for sets of cooperating ion transporter paralogs would be adapted to function together and would be favored by natural selection as a group in response to environmental change. So the role of epistasis in promoting adaptation was debated during the evolutionary synthesis and has remained controversial today. And as you recall, Sewell-Wright's shifting balance theory placed paramount importance on allelic combinations or epistasis as the main substrate for selection, fueling adaptation. And our study is the first empirical study that demonstrates that positive epistasis could promote rapid parallel adaptation on a polygenic and genome-wide scale. And so the mechanisms that we observed here might be important for driving rapid adaptation in response to environmental change, and it might be much more common than previously appreciated. And with that, I'd like to thank all my funding sources, my lab members who I cited, and my collaborators. Thank you.